Olson. We are going to be talking about reef fisheries, and our first presentation is going to be virtual, and we're going to be hearing from Brice Semmens from Scripps Institute of Oceanography and also from the Reef Environmental Education Foundation. And he's going to be talking about a 30-year retrospective using citizen science data. And yep, go.
Okay, hi everybody. My name is Brice Semmens, and I'm going to be talking about a retrospective analysis of grouper trends in the Caribbean using citizen science data. The species that we're focusing on today are the tiger grouper, black grouper, red hind, Nassau grouper, yellowfin grouper, and to a lesser extent, Atlantic Goliath grouper. Each of these species are medium to large bodied mesopredators in Caribbean coral reefs. They're culturally and economically important. They also play an important role ecologically on Caribbean coral reefs. And so we do our best to manage these species for all of those reasons. But by and large, the species aren't doing particularly well throughout the Caribbean basin. Most are either threatened or near threatened or vulnerable. In the case of tiger grouper, with IUCN listings, it's listed as data deficient. And only among these is red hind listed as least concerned by the IUCN. These are region-wide assessments at the Caribbean basin scale by the IUCN. And the reason why a lot of these species are not doing well is because there are myriad threats to the species, most especially harvest pressure that happens when these species form spawning aggregations. Most form predictable spawning aggregations that are once found easy to target because of their predictability in both time and space. And this targeting ends up resulting in effectively serial repletion throughout the Caribbean as these aggregations are fished to exhaustion one by one. Coincident with overfishing, these species tend to be relatively longer lived and large bodied, and as a consequence, they are not as able to bounce back as quickly as some more rapidly growing species. At the same time, the region is undergoing climate threats, so that there's somewhat rapid changes to things like degree heating week, and there's plenty of reason to suggest that these environmental impacts are also going to play important roles in the population trajectories of these species, especially in the early life history, post-gamete release, when the larval forms of the species are floating around in the deep briny blue. And so we can use information potentially about both impacts from fishing and human communities, and also changes in climate, to say something perhaps about how these populations might be trending through time. In order to do that, though, we need to have fairly high resolution data that's both spatially and temporally representative. So moving beyond an IUCN type red list assessment, where its assessments are carried out at the regional scale, and trying to tackle this at the level of individual countries. And in order to do that, we have to have a rich data set. And here today, we're arguing that that rich data set actually takes the form of a citizen science survey project called Reef's Volunteer Fish Survey Project. And the neat thing about the Volunteer Fish Survey Project, it has been going on since around 1993, and it's got relatively broad spatial coverage throughout the Caribbean basin. One could easily argue some of the most comprehensive spatial coverage of any consistent fish survey anywhere in the Caribbean basin. And also, a lot of these areas that are surveyed are repeatedly surveyed across years, and within years, lots of survey effort happens. And because of that, in those locations that are relatively rich in their survey effort, we can actually drill into particular countries and ask questions about how change in these communities might be transgressing over time. And we're going to do some filtering to identify those particular countries for which the data are sufficiently rich to do this type of a modeling exercise. And our criteria for filtering the data is only identifying those and using those those data from countries that had at least 1,000 surveys from the Reef Fish Volunteer Survey Project over the last 30 years, that only had 5 of those 30 years missing or less. And then finally, where at least 100 different surveys cited that particular species. And so we can whittle down those particular countries and species for which we have sufficient representation in order to carry out these analyses. And in order to analyze these data, we're going to use a spatial temporal modeling approach, and we're doing so within a framework called SDN-TNB, or Species Distribution Modeling Template Model Builder. This is a fairly well-trodden spatiotemporal modeling tool that allows us to account for effectively autocorrelation in both time and space, and importantly allows us to drive the bus in terms of changes in population trajectories as a function of effects within our model. So in our model, we're going to assume sighting probability, the probability of an individual reef surveyor detecting an animal at least once during a survey, is a proxy for abundance of that animal on a home reef. And as fixed effects to drive the changes that we might see beyond simple ups and downs of a population, we might expect that things like the MPA status might affect regional representation of abundance in these animals. Also, fishing threat as proxied by market gravity, or effectively the amount of humans nearby. We're also going to look at climate change and specifically fit an effect for sea surface temperature anomaly, and then degree heating weeks above 4 degrees C. And then we also have information on habitat through TNC's regional habitat GIS database. So we're going to use habitat type, depth, and bottom temperature as all environmental covariates. And finally, we can incorporate observer effects like bottom time and surveyor expertise. And skipping to the end, let's look at some of the results from the Bahamas and the Cayman Islands following this modeling exercise. First, the Bahamas. This is the trajectories of those different species in terms of mean percent probability of occurrence across time, starting around 1993. And I think what you will notice quickly is that while most species are declining probably a little bit, it's Nassau grouper in particular that appears to be really declining rather significantly from the Bahamas. And if we look at the Cayman Islands, we see a similar result where there's not a lot of change. If any change, most of the species are tending towards decline. Here, though, Nassau grouper are not showing what we would probably expect for Nassau grouper to expect in the Cayman Islands based on all of the previous work from the Room Project. That is, we would expect increases in Nassau grouper from this region. So this is quite curious. The nice thing about this analysis, though, is we're not just looking across the entire countries, but we're doing a spatial analysis so that we can actually look at what components of these different countries is driving that change. And so let's drill just down into Nassau grouper and look at Nassau grouper changes in sighting frequency from the beginning of the time series to the end of the time series. And what we can see is there's some fairly dramatic declines in the Bahamas, especially in the south, in terms of sighting frequency from early in the time series until 30 years later in a more contemporary context. That sighting frequency is particularly dramatic in the south. And if we change that into a rate of change where red means a greater decline and sort of a blue or green means a greater increase, what we see is that, yeah, there are fairly significant declines in the south. And the only places where there appear to be any increases through time is next to marine protected areas and also those locations that are relatively far away from human communities. And if we look at the effects within the model of our environmental drivers, that plays out, where we see sea surface temperature and market gravity in the Bahamas is particularly important for driving changes in abundance. And if we look at Nassau grouper, it's market gravity, the presence of nearby humans as a proxy for fishing pressure that is very clearly having a large effect in terms of where the spatial trends are in terms of decline in the population. If we jump over to the Cayman Islands and we look at the spatial distribution of sighting frequency in the early of the top versus the late sighting window of time of the 30 years of observations, what we can see is that there's a relatively dramatic change in the sighting frequency across time, but it's really focused on the sister islands of Little Cayman and Cayman Brac, which is, of course, where most of the work from the Grouper Moon project takes place. And if we actually turn that into a rate of change through time, what we can see is the island of Grand Cayman is having a relatively consistent decline throughout the island's coast shelf in terms of change in sighting frequency, decrease in sighting frequency over time, versus the two sister islands of Little Cayman and Cayman Brac, where we see increases. So when you put it all together, it doesn't look like there's much of a change in Nassau grouper. And it's only when we split out the sister islands against Grand Cayman, the large island where all of the populace is, human populace, that we see, okay, yeah, clearly there are rather significant increases in abundance of Nassau grouper. It just so happens that they're only happening at these two smaller sister islands. And of course, this plays out for Nassau grouper, where we see within MPAs, which happen to be very consequential in the sister islands, and in the one location in Grand Cayman where we see increases in MPA. So it's not surprising that MPAs are having a fairly large effect on Nassau grouper. And then just included down here a habitat effect, just to show that in the system, Nassau grouper are very much found in shallow habitat, which perhaps is not a surprise for most of us. They are very good at hunting in the shallows. Okay, and if we look across all of the countries and we look at the relative abundance, we'll mean occurrence as a proxy for abundance, we can see that Turks and Caicos and the Bahamas in general, in the most recent sighting period, have the most abundance of Nassau grouper in terms of relative to other countries, with Caymans and Virgin Islands falling towards the back end of this, with British Virgin Islands least of all, even less than, surprisingly to me, the United States, basically Florida Keys. And then if we switch over to the right here, we look at the change in occurrence from the early window of time to the late window of time. And it is the case that in general, those areas that have the most Nassau grouper also appear to have the steepest declines in Nassau grouper. And this is largely probably due to fishing pressure, as we discussed. So if we can actually put both these on the same panel and create sort of a four-quadrant plot where we have on the y-axis here change in occurrence probability, and on the x-axis here occurrence probability in the most recent period. Here we would expect high occurrence probabilities recently, and high increase in occurrence probabilities going to be up here in this upper quadrant. No place is. None of those countries are. Here we've got islands that, or locations, countries that are in relatively high occurrence probability, but these also happen to be in general the ones that appear to be, if anything, showing either stability or rather dramatic declines, and this is especially the Bahamas and the Turks and Caicos. So that's kind of not a great news story. The better news story is that for those areas that don't have a lot of Nassau grouper left, like the Virgin Islands, British Virgin Islands, US Virgin Islands, and the United States, there's plenty of evidence to suggest that in fact, probability of recurrence is relatively stable in these areas. So they're not declining further, which is good news. And there's some evidence to suggest that within the United States and the US Virgin Islands, there is some recovery ongoing, and this of course tracks with some of the work that is happening in the Virgin Islands with Recreational group at all. So interesting story and a mixed bag, and of course it really matters at the end that we account for the fact that space matters as well as time when we're assessing these populations. So with that, I just wanted to say thanks and give a special thanks to all of the volunteer divers that have spent hours of time underwater collecting these observations so that we as scientists can interrogate them, to say something useful and informative for fisheries management. Without them, we wouldn't be able to do the types of analyses we can. So with that, I'll say thank you, and I should be, in theory, online to answer questions. All right, thank you.
Okay, I think we can have one quick question for Brice if we can get him online. Is it happening? Okay, we'll get the question. Hopefully he can hear and answer if anybody actually has a question. Oh.
Hi, Brice. I'm just curious, how do you incorporate sampling effort into this model, or is that something that it's already integrated in?
Yeah, I think that's Henri. I can't see you, but I can hear your voice. So part of what we do with the sampling effort is we do some filtering to make sure that we have reasonable representation within country and across years and somewhat spatially. So we do some pre-filtering to make sure that we're only using those locations for which we have decent data. And then beyond that, you're correct that as a citizen science data, we can't tell people where to go collect data. So we're sort of at the whim of where people want to go diving. We buffer against that at least a little bit by including a spatial autocorrelation term. And this effectively acts somewhat like an index standardization where even though we have very unequal sampling effort across locations, the fact that we have autocorrelation within our spatial sampling frame within the model allows us to borrow that information across locations and deal with that to some extent. It doesn't completely solve the problem, but it does a lot. It does a lot to address the problem of unequal sampling effort.
Thank you, Brice. I think we need to move on. And so our next presenter is going to be Max Titcomb, also from Scripps and also using a very similar type of data. And this time we are moving to sharks.
Hello. Setting a timer for myself. Hi, I'm Max. I'm from Scripps Institution of Oceanography. So I work with Brice. And I'm excited to be here at my first PCFI. So today I'm going to be talking a little bit about some of the work I've done alongside Brennan Talwar and a suite of lovely people to similarly leverage citizen science data to help answer these questions about population dynamics and some regional success stories for Caribbean sharks and rays. And so similarly to the last project, the goal of a lot of this work is to better support and inform a lot of the good work already going on in these countries. And so we're just going to be providing a few case studies, but I would be happy to talk more about any of the areas that we've modeled with any of you if you're interested. So this is a recap for a lot of us, but we've lost about half of all sharks and rays since 1970, mostly from overfishing. Around 80 million sharks are killed per year, and 30% of those species are threatened according to the IUCN. And in addition to that, a lot of these species exhibit vulnerable life histories, so they only produce a few pups per year. They grow slowly, they reproduce slowly, they can't bounce back from highly extractive situations super quickly. And so with this knowledge that on a global scale, we're having sharks absent on about 20% of reefs, and that's a trend that also holds regionally. We're interested in taking that statistic and applying that to management somehow. And so how can we use these kind of global metrics, these regional metrics from the IUCN to better inform how like countries can be pulling data in? And so you can see across there's a lot of EEZs throughout this region. And so there's a whole suite of different management techniques being used. And so helping provide data specific to your countries is something that we're really excited to try and help with. And the way that we did that was both helping resolve regional data gaps, where can we provide new sources of data where there haven't previously been population assessments, and helping rebuild population or helping build country level trends similarly to the grouper work previously. And we're able to use that, again, using citizen science data. The Reef Environmental Education Foundation is amazing and has a lot of spatial and temporal coverage. And for this project, we had to do a bit more extensive data cleaning to help adjust for some of the variability and low sightings that we see in sharks relative to groupers. And so even after all of that, we still have 140,000 surveys across the Caribbean that we're able to incorporate into this analysis. And using that data, I'm going to be talking about 3 of the species that we worked with. ranging from endangered to least concern on the IUCN listings. So I'm going to focus in on the Caribbean reef shark, the Atlantic nurse shark, and the yellow stingray. And the first two of those are target fisheries in some countries, and the yellow stingray are a non-target species. And so we're able to look at a range of different elasma ranks using this data set, which is really cool. And the IUCN assessments for these species pulled a few different data sources for these statuses to be updated. And we're interested in just getting that number up. And so this is the approach that we took to do so. So looking specifically at sighting frequency across a range of Caribbean countries, this is for all reef-associated sharks. We see that for areas that we have low survey effort, medium survey effort, and high survey effort, those countries on the left side of those boxes are where we see higher likelihood of seeing elasma brinks. And so while we can compare sighting frequencies between these countries, especially the medium and high survey effort places, we can really only build really robust temporal trends using some of these high survey places. And so that's what I'm going to be talking about today. And those are the countries that we have highlighted in green here. And for this presentation, I'm going to focus in on Belize in the United States. And so those kind of highlight different stories related to a mixed use fishery and also more recent full protection within the States. And so we're going to be kind of walking through those step by step. And similar methods to the last presentation, we were able to integrate these all of our data into what's essentially a generalized linear mixed effects model that includes spatial and spatiotemporal random fields. And so our goal just broadly is what's the probability of someone going out and seeing at least one of our species on a dive? That's what we're modeling. And we're able to do that by using these spatial random fields, which help adjust for a lot of the observer effort issues of like, if you have a lot of surveys at a given time and a given site, it helps adjust for that. And it also helps pull and borrow strength from regions with high data to surrounding areas with lower data. And we're also able to fill in gaps. So if you're missing a year or like you have less surveys in a given year, it's pulling from surrounding time steps. And we're using, again, a set of similar covariates. And so jumping into the Florida Keys, we have over 24,000 surveys across 30 years. And so the data set starts in 1994 for this analysis. And this is just after the fishery management plan was implemented for the Atlantic Ocean within the states. And so we're able to look at immediately after that, what have populations been doing? And so this is specifically the nurse shark. And so this is our estimated sighting probability through time on the right-hand side. And you can see that it starts off as an upward tick, goes down, and then upward ticks again. And just beneath the image of the nurse shark, there is our density curve of estimates. And so if it falls to the right-hand side of the line between the last 3 years and the first 3 years of the data set, there's been an uptick in occurrence. And we see that when we actually incorporate our trends into a Mann-Kendall trend test, that there's been overall stability in the population. And when we look at the raw sighting probabilities just from the data, so just the number of sightings over number of surveys, we see that our models align pretty well with that. And that's a quick sanity check to be like, are we hallucinating things that aren't truly happening, in addition to a lot of other model validation tools. And when we actually show previous work on top of this nurse shark trend, we're able to do another sanity check of some of the work that Dan Greenberg et al. Did in 2024. And it shows that reef data for nurse shark in the Caribbean really match up well with NOAA more like systematic surveys, dive surveys that they've been doing. And we're able to validate our models by comparing to this previous work and also add on a few years of recent increases as well. And when we specifically look at reef sharks, we see that there's, since a protection in the States in 2006, we've seen kind of an upward tick in population from 1% in the first 3 years of the data set to 6% sighting occurrence in the last 3 years. And so that's a success story and shows a sign of marked increase. And when we compare these two target species to a non-target species that is not where we don't have a fishery, like they're not being targeted extensively, Ward Page et al. 2010 showed that up to 2007, there was a precipitous decline of this yellow stingray, and that our updates after that have shown a slight increase, but that overall we've still continued to see declines and an inability to rebuild that population. And so that was our case study for the United States. And then in addition to that, I'm also going to be talking about some of our results in Belize. So we have about over 3,000 surveys for this area. And in addition to Glover's Reef, we also have quite a few other places being sampled. And while we don't have the same number of surveys that we would have for the Keys, it provides, like this data is still providing consistently enough through time data for us to build some of these trends, which is really cool. And so when we look at both nurse shark and reef sharks here, we've seen marked increases. And they've had a range of shark fishing and a bunch of recent protections. And I think this is a testament to some of the conservation work and the ecotourism that has been talked about in previous presentations. Yeah, so in 2021 and 2020, there were some fishing bans placed around 3 atolls and a lot of this emphasis on ecotourism. And so I think this is really a sign of success. And FinPrint, which is an awesome program, correlates well with some of this recent data and has data going back to 2009 for Glover's Reef. We're able to extend that data set spatially and also back through time to really highlight the value of the conservation work going on there. And so in addition to the work that I just showed, we've been able to produce 40 new time series across these 5 different species for a suite of different countries throughout the Caribbean. Overall, yeah, we're in the midst of prepping this for publication. And so any further insights into this data, or if you have an interest in seeing it preliminarily, I would be happy to share and would love to gain some more insights. But yeah, thank you for like all the work you've done and for the conference. I'm really delighted to have been able to present. I want to thank the National Science Foundation and the Reef Environmental Education Foundation and the people listed here. Thank you for your time.
Okay, questions for Max?
Hi, Max.
Hi.
Sorry. Which was your objective of your work in the first place? Because I was a little bit lost because you talk about surveys when probably are sightings, because probably the sightings are done by volunteers, or that's the part I didn't understand.
Yes,
Sorry.
yeah. So the Reef Environmental Education Foundation, their volunteer fish survey project, it's citizen science, but they're actually going out and doing surveys. And so it's not just like a single sighting from like a single dive. When people go out on those surveys, they get in the water, they mark down every fish that they see, and that data is then converted into a presence and absence data frame that is then incorporated into this larger data set through time. And so those presence and absences from the surveys that these divers collect are true, are incorporated into the model as true presence absence. So that's what I meant by surveys. Yeah, does that answer your question? Yeah, and I think the overall objective of this work is to both improve the number of data streams going into the IUCN assessment frameworks and provide some sort of population trends for countries that did not previously had it. And so for both reef shark and Caribbean sharks, for both Caribbean reef sharks and Atlantic nurse sharks, we were able to provide some places that hadn't previously had population assessments done.
And one more question.
Yeah.
Why you decided to use those 3 species? Because there are other species, more commercially important species. For instance, for instance, nurse shark is not so commercially important compared to the other one. Sorry.
Yeah, definitely. So the reason that we chose the 5 species shown here is that they were the most representative across the highest number of countries. And so it's kind of a middle ground of like We have IUCN regional assessments. We have a lot of people on the ground doing fieldwork in specific countries. How can we do the most amount of high resolution population reconstruction for the most countries? And so we also have like, we're kind of getting at like, what are some regional trends across countries that have enough data for us to do models? And so I think especially for Caribbean reef shark, across a suite of countries, we've seen increases. And so I think that's a really exciting story for and testament to the value of the management going on throughout the Caribbean for reef shark.
Don't look so happy, Joanna. Hi, Max. Interesting. I'm curious. Yellow stingray aren't targeted, but they have the big drop population, at least in the United States, and they're not really bouncing back very well. Any ideas why that might be?
Yeah, well, one of the reasons is I think it's a combination of both like habitat degradation, and also there's an increase in predators. So I think with this uptick in sharks, we're also seeing a decline in some of their prey species. That's not something that we've that's just conjecture, but I think based on the trends that we've built, that's my thoughts.
Do you have enough data for both of those factors to perhaps sometime integrate that into your model?
Yeah, I think that would take a different modeling approach, but I think that would be looking at like predator-prey interactions with the reef data set, I think is a super valuable step forward.
Habitat?
Sorry?
And what about habitat degradation?
Yeah, so we included habitat as like a fixed benthic habitat map to help just like adjust for areas, because we are getting surveys primarily from reef areas. And including habitat in our model helps improve the estimates for undersampled areas. We haven't incorporated changes through time in habitat within the modeling approach, but I think that would be really cool.
Okay, yes, we're going to take that up at the coffee break. Thank you for lots of questions, Terry. Thank you,
Thank you.
Max, for your presentation. Okay, next up we have Alyssa Pagel from University of Southern Mississippi, and she's going to be talking about movement patterns and site fidelity of Sheepshead.
All right. Good morning, everyone. My name is Alyssa Pagel, and today I'll be presenting on the movement patterns and site fidelity of Sheepshead in the Mississippi Sound and surrounding waters. If you have not seen or heard of a sheepshead before, this is what the species looks like and who I'll be talking about today. So a little introduction into the species. Sheepshead are an estuarine-dependent species found throughout the Western Atlantic from Nova Scotia all the way down to Brazil, including the northern Gulf of Mexico. They support both commercial and recreational fisheries, yet surprisingly little is known about their seasonal movement and habitat use, especially in the northern Gulf of Mexico. So a little bit about our study area. The Mississippi Sound is a large estuary bordered by Louisiana to the west and Alabama to the east. The northern Mississippi Sound has 3 subsystems, and to the south, it has a series of 4 barrier islands that separate the sound from the open Gulf of Mexico. These features together create a mosaic of habitats from freshwater inflows to near marine barrier environments that fish can move among throughout their life history. Within the Gulf of Mexico, management occurs at the state level as sheepshead occupy near shore and estuarine habitats throughout most of their life history. In Mississippi, current sheepshead regulations are 15 fish per person per day, with a minimum size of about 14 inches or about 35.5 centimeters total length. Over the past few years, recreational targeting has increased, driven slightly by social media and the ease of catching sheepshead around piers and seawalls, as these fish are often structure associated. Restaurants have also began serving sheepshead marketed as bay snapper, which has increased their popularity as well. This increased pressure has raised state concerns about localized depletion. So to address this concern, this study was born, and our objectives are to assess movement trends of tagged sheepshead, assess site fidelity, that is, if individuals remain or return to a given area, and evaluate connectivity among the estuarine subsystems of the Mississippi Sound and adjacent coastal waters. To achieve these objectives, we captured and tagged 61 sheepshead using anchored gill nets and hook and line gear between the years 2021 to 2024. Each fish was measured, externally tagged, and implanted with a Novasi V13 acoustic transmitter. These tags are about 2 centimeters in length and have a battery life of about 2.5 years. For those unfamiliar with acoustic telemetry, each transmitter emits a coded sound, pulse or ping, that is detected by underwater receivers when a fish swims within range. By combining detections across time and space, we can construct who, is, where, and when, essentially providing a digital footprint of an individual's movement. Our study used a collaborative network of these receivers that spans the Mississippi Sound and adjacent waters from Louisiana to Alabama. Each point on this map represents a receiver, and the different shapes show which agency maintains it. This broad coverage allowed us to track fish even as if they left our immediate study area. We then classified these receivers into subsystems to help generalize movement over time. So we'll first start by introducing and classifying our 2 capture systems. So St. Louis Bay is shown here in blue. It is the westernmost subsystem in Mississippi Sound. Here we captured and tagged 25 of our 61 tagged individuals. Then shown here in green, Pascagoula River Estuary is the easternmost subsystem in Mississippi Sound. Here we tagged the remaining 36 individuals. Other receiver groups include Lake Pontchartrain here in red, Pearl River in lilac, Lake Bourne in light pink, the Western Mississippi Sound in yellow, Back Bay in dark purple, the Barrier Islands in this hot pink, Chandeleur Islands in orange, offshore sites in dark blue, though notably we didn't detect any sheepshead at any of these sites, Grand Bay in this medium purple, and then Mobile Bay in dark green. These colors will remain consistent throughout the talk so that you can visualize movements in areas over time. And then lastly, for our methods, this bar plot shows the size distribution of our tagged individuals, where the x-axis is total length and the y-axis is frequency. The blue dashed line marks 50% maturity of males, and the pink dashed line marks 50% maturity of females. The black dashed line shows Mississippi's keeper size. And as you can see, it's very close to the 50% maturity for these fish. So harvested fish are most likely spawning capable or soon to be. Similarly, most of our tagged fish were at or above maturity threshold, meaning they were likely spawning capable or again soon to be. This is helpful to keep in mind for our analysis coming up. You might note that there was one smaller fish tagged. It did die shortly after we tagged it, so it will be excluded from our analyses. All right, and with that, let's jump into our results. This figure is a bar plot showing movement trends generally. This plot is faceted by capture system with St. Louis Bay on the left indicating westernmost and then Pascagoula on the right or the east. And individuals are on the y-axis. The bars are, the shaded one is gray to indicate how many individuals were tagged in that system in total. And then the turquoise bars in the middle are individuals that left their capture system at any point. And then the darkerish kind of green bars indicate individuals that returned to their capture system at some point after leaving. So we saw that roughly half of all tagged individuals left their initial capture system, 29 of 61 overall. We also found that several returned afterwards, indicating potential slight site fidelity. Some even moved into other inshore systems. So in St. Louis Bay, we actually saw that one fish returned to a different inshore system that was not St. Louis Bay. And then in Pascagoula, we saw the same, but with 4 fish. We found no size differences between the fish that moved and the residents, but system level differences were kind of apparent. Pascagoula fish tended to return more often than St. Louis Bay fish. To analyze this further, we looked at residency indices to assess and quantify site fidelity. Residency is essentially the proportion of days a fish was detected within a system. In this box plot, we're looking at residency during spawning and non-spawning months. Here we have season colored on the x-axis, residency index on the y-axis, and the plot is again faceted by system, with St. Louis Bay on the left and Pascagoula on the right. In both systems, residently declined significantly, both figuratively and statistically, during spawning months, shown in the yellow-orange. These patterns are consistent with seasonal offshore movement associated with spawning migrations. That said, residency during spawning months isn't zero, even though not some or most, if not all, of our tagged individuals were potentially spawning capable or about to be. To illustrate these movements at the individual level, here's an abacus plot of a few of our tagged fish. This plot has date on the x-axis and transmitter or individual ID on the y-axis. So that means that each row is a unique individual. Each closed point is a detection of the fish, and the colors indicate system that they were detected in, which you can see in the key, and then visualize in the map above as well. The open circles are the date that a fish was initially captured and tagged, and then the X's on the plot are date of estimated tag death. That's the battery life of the tag. The gray shaded bars, which you can't probably see, indicate spawning months. And then lastly, we have numbers on the right side of the plot that are the total length of the fish at time of capture. So let's focus on a few of these examples. This fish from Pascagoula River estuary is an example of one of our resident fishes. This is a fish that never left its capture system. We did tag it at 386 millimeters, which is over both male and female maturity marks. This fish also would have grown over time. So even if it didn't make that spawning run the first spawning season, we would have expected it to make it at either the second or third spawning season that we saw it tagged. In contrast, these fish all made spawning runs. So you can see all of the pink dots that are in there are movements to the barrier islands. This is consistent with spawning runs and what we would expect to see from these individuals. And then we also have a few examples of fish that left their capture systems and then never returned. These fish all actually moved to different states. One did return back to Mississippi, though the other two did not. So this really just helps us visualize the range of movement strategies that we observed from resident to migratory individuals. To summarize subsystem transitions, we built movement networks. Each node represents a subsystem, and then node size reflects centrality or how connected the subsystem is, and the edge thickness or the thickness of the line shows how many transitions occurred between two given subsystems. Again, on the left, are fish tagged in St. Louis Bay, and on the right are fish tagged in Pascagoula. This plot is only showing individuals that left their capture system at a given point. We see a trend that St. Louis Bay tagged fish tended to stay near subsystems to the west, and then fish tagged in Pascagoula overlapped a little bit, but tended to stay more east. We did, however, see that both capture system fishes were using the barrier islands, which really helps us understand and see that the islands appear to be key movement corridors and connectivity hubs, particularly during spawning months, though these plots show movements across all months and years. This is a plot of those two plots together. So this is all fish that left their capture system together. If you have any questions about it, come see me after. Overall, we saw seasonal spawning movements are clear, temporally consistent offshore directed movements during spawning months, February to April for some individuals, though other remain inshore and even in system year round. We saw partial site fidelity. Many individuals returned to their original systems after running and making spawning moves, though some do not. Barrier islands also act as connectivity hubs. Most migrants use these habitats at some point, really underscoring their ecological importance. All right, quickly stepping back, these results reveal a more flexible movement ecology than previously recognized. Many presumed spawning capable individuals left inshore areas to spawn, but some remained in estuaries, suggesting either inshore spawning, delayed migration, or skipped spawning years. From a management perspective, the moderate site fidelity we observed means populations could experience localized depletion, especially around high pressure inshore structures. Yet cross-system movement and larval dispersal likely maintain regional connectivity. For future research, we plan to expand tagging efforts to the barrier islands and around the Gulf to better understand their movement as a whole outside of those two systems. In summary, this is the first system-wide assessment of sheepshead movement in Mississippi waters. We show that sheepshead exhibit seasonal offshore migrations, moderate site fidelity, and partial connectivity among estuarine subsystems, and findings will help inform management. With that, I'd like to thank my acknowledgements, especially the GCFI Ron Schmid Scholarship. And with that, I'll take any questions.
Of course, Terry has a question.
Gee, that was fun. You say partial site fidelity, and usually that means, from what I'm getting, that fish go out to a spawning aggregation site, turn around and go home, and no one's rifled the cupboards. They go right back to where they were from. What about spawning site fidelity? Do you have any data on that?
No, so there's actually no data on where spawning is actually occurring in Mississippi. It's thought that they go offshore and that they use the barrier islands as corridors, but there's been no evidence of where exactly that's taking place. We don't know that yet.
Nice presentation. I was about to ask the same question, but how many fish you already got? 36 fish only, and I remember, and you mentioned the numbers and a lot of receivers, and those receivers were basically recording the presence of the fish. And your key objective was just to identify how the fish move, but you mentioned the spawning, and it's important to know where those fish are actually spawning.
Mm-hmm.
That was part of your objective, or just to know how they are moving?
Really just to know how they're moving. We would really ideally like to know where they're spawning, and that's just not something that we've been able to achieve yet. But with the acoustic telemetry, our goal is to just understand where they're moving, and how they're moving.
Hi, great presentation. You mentioned inshore and onshore. I mean, my question is, what environmental or behavioral factors do you think is driving that? Do you feel that the acoustic coverage was adequate, first of all? And do you think what could be influencing or driving these movements, and what management do you think should be applied, would be my question.
Yeah, so that's our very next step to look into, is what environmental drivers are driving their movements offshore. We're going to look at temperature, tidal influences, and river discharge, things like that. Yeah.
Also, just the type of activities, because I think you'd mentioned they like to hang out near piers and structures. Are you going to be looking at, or would you suggest that be included as well?
Sorry, could you repeat?
Looking at actually the physical structures, what do you think? Are they influenced by development?
Oh, yes. So they use structure often to feed. They feed on the barnacles and such around the structures. So that's why we tend to see them there. We see them feeding, and that's often just an easy place to target them.
Okay.
Did I answer your question?
Yeah, I think we need to move on. Thank you, Alyssa. Next up, we have Jack Olson, who's also going to be talking about acoustic monitoring, sorry, but using it in a slightly different way.
All right. Can you guys hear me all right? All right. Yeah, so I'm Jack Olson. I work at FWC in the Florida Keys, Florida Fish and Wildlife. And today I'm going to be where we're using acoustic telemetry in marinas in the lower Florida Keys to try and identify fishing mortality of our tagged fish. So this project is part of a larger research effort centered around the Western Dry Rocks seasonal closure. So it's a seasonal no-take zone. And this site, it's a offshore reef system located approximately 17 kilometers southwest of Key West. And it's home to multi-species spawning aggregations of snapper, grouper, and jacks, among other species. And for this reason, it became heavily targeted by fishers up until 2021, when FWC decided to enact a seasonal spatial closure. And the closure is relatively small. It's 1 square mile and lasts from April through the end of July. And this period was designed to protect spawning snapper primarily, and also jacks, permit namely. So since then, our team at FWC has been conducting monitoring to assess the effectiveness of the closure to protect spawning species. And specifically, we're looking at spatial movements of snapper using acoustic telemetry. We're also assessing age growth and reproductive trends through life history sampling. And then we're also estimating changes in densities and length compositions with a combination of visual census and electronic equipment such as cameras. So I mentioned acoustic telemetry. Since 2021, we have tagged a total of 121 gray snapper and 119 mutton snapper. And these are tagged with acoustic transmitters that are surgically implanted. The top right is a picture of a nice mutton snapper we caught, hook and line. So we do all of our tagging topside, and the fish is kept in a bin like this in water, and the surgery is done, and fish goes on its way. These fish were released within a broader seascape array in the lower Keys composed of 76 VR2W and TX receivers. And these are among other receivers we have throughout the Keys, stretching from the Tortugas up to Key Largo. The goal of this tagging was primarily to assess movement pathways to and from the spawning site at Western Dry Rocks, but also to look at where and when these aggregations were taking place specifically, because that has, it's important for assessing the effectiveness of the reserve, seeing if it's actually protecting the aggregation itself. So in addition to the movement data, we are getting fishing mortality information through a tag return program with angler rewards. So anglers are given a $100 gift card if they return an acoustic tag to us. So despite this tag return program, we wanted to know were some of our fish being caught and harvested, and we didn't get a call. So we wanted to look at unreported harvest. And this is where marinas come into play. So we kind of had an idea that maybe some of our tags might be ending up near fillet stations and marinas in Key West. So we started a pilot survey using a VR100 hydrophone you see on the bottom right to see if there are any tags in these marinas. And lo and behold, we found a few, and these are stationary tags that were in multiple marinas. So those were fish that were harvested and the tags are there. So based on this, we designed an acoustic receiver array targeting several marinas in Key West to continuously monitor for these harvest incidences. Yeah, and just want to note, these discard locations are really predictable both in time and space. As you can see in the top right, the scavenging predators certainly know where to be and when to get a free meal. So we're able to use that predictability to our benefit. So before we designed the array, we set up two core objectives. The first was to quantify unreported harvest of tag snapper just through acoustic monitoring of carcass discard locations. And the second was to characterize any habitat utilization that might be happening by either gray snapper or mutton snapper. So we designed an array targeting 4 marinas covered by 13 total receivers. These receivers were deployed from July 2023 to July 2025. And within each marina, there were 3 receivers within the core area and 1 receiver in each marina that acted as a gate and was in an entrance channel to detect the ingress or egress of tags. So we really wanted to separate out tags that were swimming into a marina to tags that were being carried in by boats. After we got our 2 years of detections, we wanted to come up with a standardized way to assign fates to individual tags that were detected within the marinas. So we came up with a decision tree basically to separate into 2 fates, alive and harvested. And we took into account criteria such as movement between stations, both within the marina and also in the seascape, the amount and timing of stationary detections on any one receiver within a marina, and then also the detection history of individual tags prior to arrival in the marina. So for instance, if a tag disappears from the middle of the seascape array and then suddenly shows up in the marina, that tells you something different as opposed to a tag that is sequentially heard on multiple receivers all the way into the marina. And just quickly, regarding the stationary detections, we calculated behavior thresholds from the full suite of tags in the seascape to try and get maximum amount of time a given tag in a live fish could be expected to be stationary, and we separated it out by species. So so far we've heard 13 gray snapper tags, 7 of which were live fish, 4 were deemed harvested, and 2 were unknown. So the unknown tags were basically tags where we had very few detections on a single receiver, and we weren't confident in making any kind of a fate assignment on those. Of the mutton snapper, all 5 were deemed harvested. And so interestingly, of the gray snapper, we see habitat utilization throughout the year. In the summer, we saw them making movements from the reef and to the marina back and forth, often multiple times over the course of a single summer. So they're heading out to the reef to spawn, going back to the marina, and doing that month after month. And yeah, it's not particularly close out to the reef site. Average one-way distance was 16 kilometers. And in contrast, none of our tagged muttons were using marinas as habitat. So speaking of mutton snapper, I just want to highlight one fish as an interesting anecdote. This fish was tagged by us in May 2023, mutton snapper. And yeah, we tagged it topside at the aggregation site around the full moon. Over the course of the next year, it was detected up the coast of the Keys, up as far as Marathon, potentially further. That's just as far as we heard it. The next spring, we hear it making its way back down, back to the spawning site of Western Dry Rocks. So over that course of that year, it traveled at least 160 kilometers and potentially further. Just Marathon was as far up as we heard it. So it arrives back at Western Dry Rocks for the full moon. And shortly after it arrives at Western Dry Rocks, we hear it in the marina. And that tag is sitting on the bottom of that marina today. So it just shows, without monitoring these marinas, we wouldn't know definitively what happened to that fish. And it's really nice to have a known fate. And I just want to also mention, we were there tagging that night that fish was caught in May 2024. And this picture was taken the evening that it was caught. So potentially one of these boats actually caught the fish. So a couple of interesting points there. One, just the highly predictable nature of these aggregations. The fish know where to go, as do the fishers. And also there's some potential implications for design of the reserve if aggregating fish are still being caught. So by incorporating marina detections into our mortality estimates, including tag returns, we're able to increase our estimates of fishing mortality by 50% for gray snapper and 55% for mutton snapper. And we are also able to calculate an alternative tag non-reporting rate from how it's typically calculated. And basically to calculate this, we took the unreported tag harvest. These are tags that showed up in the marina and we never got a call, divided by the total tag harvest. So total tag harvest being the ones we got a call for and the marina tags. And for gray snapper, this value was 33% tags were unreported and mutton snapper is 36%. So another way to look at it is for every 3 gray snapper tags we got a call on, there was 1 fish out there was caught that we didn't know about. So just leave you guys with a few takeaways. First and foremost, this marina monitoring effort really increased and improved our estimates of fishing mortality, both for gray and mutton snapper. And it also gives us an alternative method for estimating non-reporting rates, which is important for contextualizing and refining fishing mortality estimates. And found that marinas were important habitat for gray snapper, primarily in the fall and the winter, but as I mentioned, they were there to some degree year round. And yeah, when you're thinking about whether an area is amenable to this type of monitoring, the fact that Key West is pretty highly spatially concentrated with precise and predictable discard locations makes it cost effective to implement this kind of array, and that might not be the case everywhere. And moving forward, we want to keep this array in place to potentially detect any changes in unreported harvest due to increasing our tag rewards. We think that could be an interesting natural experiment. And as I mentioned, we want to, in the future, use the non-reporting rates we calculated here to refine our fishing mortality estimates with or without the marine detection program. And then ultimately, this known fate information will be very important for training multi-state models of capture probability from our full acoustic detections from the seascape. Thank you very much. I just want to take a quick second to acknowledge the funding source that allowed me to come here. This is the Dave Hoptoff Fund for Emerging Scientists. Thank you very much.
Okay, thank you, Jack. Any questions for this very interesting take on acoustic monitoring?
Thank you. Did you estimate growth rates? And if the answer is positive, did you find differences between males and females?
No. So unfortunately for these tagged fish, we don't have sex. So we've subsequently started using catheters to get sex of our tagged individuals. But at the time that these were tagged, we did not have sex. Good question, though. Thank you. Yeah.
Yeah, super interesting. I'm curious, do you know what happens if these fish are preyed on by sharks or some other larger predators? And is that something that's typically looked at? Did the tags pass through the digestive system? Or are you like, something migrated 400 miles? Wow, that's an amazing snapper, where it could be some migratory shark species, for example.
Yeah, so good question. A subset of our tags are predation tags, and they actually have a coating that when the stomach acid of a predator encounters it, it changes the signature of the tag, and we can know. And none of these fish had predation tags inside of them, but we're going to be using that in the future to look at depredation.
Okay, anyone else? Great, thank you very much, Jack. So next up, we are going to have Krista Sherman from the Bahamas, and she's going to be talking about differences in stock status between traditionally harvested and recreationally harvested fish species.
Thank you. Good morning, everyone. I'm Krista Sherman. I manage the Fisheries Research and Conservation Program for the Perry Institute for Marine Science. And for the last couple of years, I've been working on a project promoting sustainable, healthy domestic seafood markets in the Bahamas. So the work that I'm going to be discussing today is just one component of that project. So I worked with some scientists that are experts in fisheries stock assessments to assess the status of traditional and recreational fishery species in the country. So we've heard quite a bit about the Bahamas this week so far, but fisheries have a really long history of providing economic, cultural, and ecological benefits to developing coastal nations like the Bahamas. So as you can see from this picture here of historic catches of sponges from the 1800s. They're also an important source of food security and nutrition, supporting the livelihoods of thousands of fishers, not just those directly involved within the fisheries sector, but as well through tourism opportunities as well. They're an important component of biodiversity in maintaining healthy and functional marine ecosystems, and they're really integral to the culture of the Bahamas. And if we have robust data to inform sustainable fisheries management, this would not only ensure the long-term viability of fishery populations, but also ensure that they continue to provide both the economic, ecological, and cultural benefits to communities as well. And this really underpins the work that we do. It's to promote sustainable fisheries management. However, managing fisheries across the various sectors from subsistence to commercial scale has proven to be a very complex challenge in the Bahamas. And this is due to its vast spatial extent, limited capacity and resources for enforcement, and that's coupled with really strong sociocultural and political dynamics. And this is particularly evident for some of the traditional fishery species like spiny lobster, queen conch, and Nassau grouper that are in heavy demand, but really vital to the commercial fisheries sector. And despite existing regulations, we continue to see overfishing and increasingly habitat degradation, which threaten these fishery populations. And unfortunately, very few species-specific regulations exist. And there's a lack of comprehensive data on stock size, which is really critical for estimating total catch. And that's important if we want to have effective management strategies. So limited to no stock assessments for a lot of these species. So to address this, we conducted stock assessments of both traditional and recreational fisheries species in the Bahamas. And we focused on reconstructing catches analyzed with a stock assessment tool that's suitable for data poor environments to evaluate fishery reference points for data limited stocks. And hopefully here we can use this information to advance sustainable fisheries management for these particular species. So the species we looked at included 2 invertebrates, queen conch and Caribbean spiny lobster, which is locally called crawfish. We looked at 8 demersal species, and these included 4 species of grouper, Nassau grouper, rock hind, black grouper, and yellowfin grouper, as well as 3 snapper species, mutton snapper, lane snapper, and gray snapper, as well as hogfish, which is a large wrasse, even though locally in the Bahamas it's referred to as hog snapper. And then we also looked at 2 pelagic species. So we had dolphin fish or mahi-mahi and wahoo. So specifically, we use this CMSY++ method. This was developed by Froese and published originally back in 2017, and this has recently undergone some refinements. I'm not going to go into the details of the method, so you can have a look at the paper if you're interested. And we updated catch reconstruction for the Bahamas that was originally published by Smith and Zeller back in 2016, and they looked at the period 2050 to 2010. We also incorporated fishery-dependent data, so landings data provided by the Department of Marine Resources from the period 1996 to 2003. With this assessment as well, we also included reports of IUU fishing and supplemented that with FAO and regional reports. We also included for fishery independent information, our survey data for these particular species between the period 2001 to 2019. So we really use this information to better estimate historical exploitation as well as relative abundance for these species. So looking at the total reconstructed catches for the country, that totaled about 1.3 million tons during this period. And the 12 species that we looked at represent about 69% of the total catch. So what you're seeing here, the solid red lines are showing you the catch trends over time. And in the sort of pinkish, reddish shaded areas, the 95% confidence intervals. And the gray dashed lines are showing you maximum sustainable yield, and in gray, the confidence intervals associated with those. So as you can see, catches for all of these species have increased over time, and in many instances are above maximum sustainable yield. For Nassau grouper, which is a critically endangered species, there's about a 40-year stretch where catches exceeded maximum sustainable yield for this species. And for Caribbean spiny lobster, which was certified in 2018 as an MSC fishery for the Bahamas, we can see in recent decades that catches are also above or at maximum sustainable yield for this species. So we assigned the status of stocks for these species relative to these ranges of biomass. So a healthy stock would have a healthy stock would have a stock biomass B that is above or greater than maximum sustainable yield, whereas a stock that is overfished would have a stock biomass below maximum sustainable yield. So looking at the results from our stock assessments, you can see that most Bahamian fishery stocks are in decline, as evidenced by the stock biomass below maximum sustainable yield. The only exception here is mahi-mahi or dolphin fish that's healthy. And this may not be surprising given the life history characteristics of this particular species, which are different from many of the others listed here, so less vulnerable to fishing pressure. A little caveat though with the data on pelagics, it's a bit patchy and a lot of this information is unreported, so these should be treated as conservative. But for some of these other species, like Queen Kong, for example, it has a really long history of being exploited in the Bahamas that dates all the way back to the original inhabitants, the Arawaks. And we did have an export fishery for Queen Kong for some time. But in terms of its life history characteristics, we know that it's density-dependent reproduction and suffers from allele effects. And with many of the other fish species here, the groupers and the snappers, they also have life history characteristics that make them extremely vulnerable and susceptible to being overexploited, particularly the use of fish spawning aggregations to reproduce. Some of them are protogenous, some exhibit site fidelity to specific spawning sites as well. So you can see that for some of those species, they are grossly overfished. I didn't pretend I'm not showing a figure or presenting this here, but we did look at exploitation rates for some of these species. So for, in particular, Queen Kong, hogfish, rock hind, black grouper, and the snappers that we looked at, as well as wahoo, although they do have low stock biomasses, exploitation rates in recent years are a little bit lower. So if that continues and with management interventions, it's possible to potentially rebuild some of those stocks. So some takeaways for fisheries managers, not just in the Bahamas, but in the region. It's important to realize that relying on commercial landings data alone can be misleading. It doesn't really capture the full picture of the resources that are being removed from the system. In contrast, reconstructed catches aim to capture the sum of all removals from extractive activities. So in this instance, not just looking at commercial fishing, but subsistence fishing, taking into account IUU fishing as well. So doing so and combining that with that information offers a more comprehensive estimate of actual exploitation. And when we combine this with appropriate qualitative and quantitative priors, like the ones that we used in this study, this can provide a really reliable basis for conducting stock assessments. So for managers, there are a couple of takeaways. There's a need for the science to and the policies to evolve with the science. So we do need some regulatory reform, but that needs to happen in a timely manner if we want to have these populations recover. Additionally, there's a need for improved data collection, both fishery dependent and independent data, especially for the pelagics where that data is quite patchy, but that applies to all species that are being harvested. Because monitoring data is really critical for us to have an understanding of what's going on, to adaptively manage, and to know whether the management interventions that we have in place are being successful and they're effective. Additionally, there's a need for interagency cooperation and cross-sectoral engagement, and those need to better align policies and actions across all of the various sectors if we're going to have any real progress moving forward. And to achieve that, we're going to need some effective communication, engagement, and cooperation across all of the stakeholders. So where do we go from here if we want sustainable fisheries? Well, in 2024, Roberts published this paper and outlined 11 actions to help us advance sustainable fisheries management. And these are really grounded in two principles. And the first is that fisheries must minimize environmental harm, support the regeneration of marine life and habitats, and adapt to climate change. That's something all of us are going to be grappling with moving forward, and we need to consider that. And fisheries must support and enhance the health, well-being, and resilience of the people and communities that depend on them, not just the corporations that are benefiting from them. So with that, I'd like to acknowledge all of the people that have assisted us with surveys between 2001 and 2019, the Department of Marine Resources for providing commercial landings data, as well as my collaborators at the Sea Around Us and Aquatics, and to the Builders Initiative for funding this project and this work. And that's just a QR code so you can learn more about the Fisheries Research and Conservation Program, and we'll share updates once this paper has been published. Thank you for your attention.
Any questions for Krista?
Yeah, nice presentation. I do have a question on dolphin fish. So tagging data has shown they move throughout the Atlantic and Caribbean to some degree. And how are you able to isolate what's happening in the Bahamas relative to what would seem to be a basically Western Atlantic stock?
Yeah, that's a good question, and it's pretty difficult. So one of the things that we looked at was available stock assessment work that's been done, particularly for those two pelagics, since they are highly migratory. You know, there's room for improvement there to be able to completely isolate that, because the data that we have in country is quite limited and patchy.
Any more? Okay. Thank you, Krista. So right, our final presentation for this session is going to be virtual. We're going to be hearing from Fabian, Pina, Amargos about the fact that stronger enforcement has enhanced the status of coral reef fishes in Cuba.
Buenos días. En nombre de los autores, agradezco a Gulf and Caribbean Fisheries Institute y Environmental Defense Fund por permitirnos compartir estos resultados científicos. Lamento no poderlos acompañar en persona, pero gracias al empeño de todos los organizadores y la tecnología, me siento entre ustedes. Esta ponencia resume una de las publicaciones científicas del proyecto Bogea Cuba. El Bogea Cuba es un himno de la ciencia durante la COVID. Tamara y yo estábamos enfrascados en la elaboración de varios capítulos para el volumen 18 Coral Reef of Cuba de la serie Coral Reef of the World, editado por Springer. Junto a una treintena de colegas acudíamos información ecológica, oceanográfica y socioeconómica de casi 400 sitios de arrecifes de coral alrededor de Cuba, ascendiendo a casi 40.000 datos entre los años 1980 y 2010. Los resultados, sin duda valiosos, están limitados por la heterogeneidad de métodos, observadores, hábitat y escalas espaciales y temporales. Se nos hizo evidente la necesidad de un muestreo estandarizado y actual a escala de país. Después de casi 2 años de preparación con la participación de más de un centenar de personas y más de 30 instituciones cubanas extranjeras, especialmente Avalon y Environmental Defense Fund, el bojeo a Cuba partió de Búcaro, casi 500 kilómetros al este de La Habana, a mediados de julio de 2023 y terminó en ese mismo sitio a inicios de septiembre. Durante los 3 meses más calientes registrados jamás en el Mar Caribe, muestreamos casi 70 sitios, colectando datos sobre las aguas, sedimentos, corales, algas, erizos, peces óseos, elasmobranquios, actividades humanas, mamíferos marinos, aves marinas, usando un protocolo de muestreo estandarizado y realizado por los mismos observadores durante toda la expedición. Los datos in situ fueron complementados con información online, publicaciones y reportes científicos previos. En el caso que nos compete, los peces, se tomaron datos de especies seleccionadas representativas de 3 grupos de acuerdo a su talla máxima: grandes, medianos y pequeños. Los datos fueron analizados con las herramientas estadísticas adecuadas. Detalles de este aspecto o cualquier otro puede obtenerse a través del intercambio por correo electrónico con los autores. La figura de la izquierda muestra la biomasa de los grupos de peces en las 7 zonas en que se muestrearon los arrecifes coralinos. La figura de la derecha, la distribución en esas mismas zonas de la complejidad estructural y el número de hábitats. La biomasa de peces grandes y medianos fue baja en la mayoría de los arrecifes coralinos muestreados alrededor de Cuba. Se observan grandes diferencias entre las biomasas, de hasta un orden de magnitud entre los sitios mejores cuando se compara con los sitios promedio. Martínez de la Reina, GQ, en las figuras, muestra los valores más altos de biomasa promedio para los peces grandes y medianos y los más bajos de los peces pequeños. Las demás zonas no muestran diferencias significativas o estas son de poca magnitud. Las características del hábitat también varían entre las zonas, pero sin patrones claros. En esta diapositiva, las figuras muestran la biomasa de grupos de peces en relación con la complejidad estructural, figura de la izquierda, y el número de hábitat, figura de la derecha. No hubo diferencias significativas en la biomasa de los peces grandes entre los valores mayores y menores de complejidad estructural. Por su parte, a niveles altos de complejidad estructural, los peces medianos muestran los mayores valores de biomasa y los peces pequeños los menores valores de biomasa. Estudios previos no han encontrado estas relaciones, presumiblemente debido a que hábitats estructuralmente más complejos brindan menos posibilidades de alimentación de los herbívoros, las especies dominantes entre los peces medianos de este estudio, al mismo tiempo que brindan mejores oportunidades de alimentación y refugio para los peces pequeños. En cuanto al número de hábitats, mayor cantidad de estos estuvo asociado a valores más elevados de biomasa de peces grandes y medianos, presumiblemente relacionado con mayores oportunidades de sitios de crianza, refugio y alimentación en un mosaico de hábitat amparado por la literatura científica. No hubo relación significativa entre el número de hábitat y la biomasa de peces pequeños. La figura muestra la biomasa de los grupos de peces relacionados con la intensidad de pesca, el nivel de protección, que significan sitios dentro o fuera de las áreas protegidas, y el nivel de aplicación de las leyes, que significan el nivel de patrullaje con respecto a sus frecuencias diarios, semanales o mensuales. Los mayores niveles de biomasa de los peces grandes se observaron a los niveles más bajos de intensidad pesquera. El nivel de protección no impactó considerablemente la abundancia de los peces grandes. Los mayores valores de biomasa de los peces grandes se observaron a niveles altos de aplicación de la ley. Los peces medianos y pequeños fueron menos fuertemente y direccionalmente impactados que los peces grandes por los factores humanos. Solo la biomasa de peces pequeños fue significativamente menos a niveles altos de intensidad pesquera. Los otros factores humanos, nivel de contaminación, cantidad de población, nivel de turismo, distancia de la costa, correlacionaron con los factores explicados anteriormente. Las relaciones entre los niveles de intensidad pesquera y aplicación de la ley y la biomasa de peces grandes es consistente con lo obtenido anteriormente con otros estudios internacionales y en Cuba, pero por el nivel de estandarización de este estudio son resultados más robustos para el ámbito cubano. Por otra parte, el bajo impacto del nivel de protección en la biomasa de los peces grandes sugiere que las áreas protegidas marinas no están jugando el papel al nivel de que ellas esperan. La poca efectividad de las áreas protegidas marinas debe ser analizada en profundidad y corregida usando las herramientas de manejo disponibles. Los tiburones solo estuvieron presentes en el 20% de los sitios. 3 especies de tiburones fueron detectadas durante el estudio: Inclimostoma serratum, Carcarinus perezi y Carcarinus falciforme. Las 2 primeras especies fueron detectadas en los Bay Remote Underwater Videos demersales y la última especie en los Bay Remote Underwater Videos pelágicos. Solo un individuo fue detectado en los Bay Remote Underwater Videos pelágicos. La figura muestra la probabilidad de observar tiburones en relación con la aplicación de la ley, la presión pesquera, ya sea tanto la intensidad como la actividad, y la contaminación. La mayor probabilidad de observar tiburones fueron niveles mayores de aplicación de la ley en los niveles más bajos de presión pesquera y contaminación. El nivel de protección no fue un buen predictor de la probabilidad de observar tiburones. Estos resultados son similares a los obtenidos para los grandes peces y las explicaciones coinciden con las de ellos. El análisis de las asociaciones de peces corrobora los resultados anteriores. Los resultados del poema NOVA indican diferencias en la composición de las asociaciones por zona, nivel de aplicación de la ley, intensidad pesquera y protección. Los diagramas de escalado multidimensional no métrico, NMDS, muestran que la posición geográfica no explica la ubicación de los sitios por zona, con zonas geográficamente adyacentes mostrando asociaciones muy diferentes. Ejemplos: SE, suroriental, y GQ, Jardines de la Reina, y zonas geográficamente distantes mostrando asociaciones similares. Ejemplo: SE, suroriental, y NW, noroccidental, y SW, suroccidental, y GQ, Jardines de la Reina. Por otra parte, las asociaciones de peces fueron similares, con niveles similares de aplicación de la ley e intensidad pesquera, y en ambos casos mostraron un fuerte gradiente en la composición de las asociaciones, presumiblemente debido a la desaparición gradual de las especies grandes a medida que aumenta la intensidad pesquera y disminuye la aplicación de las leyes. No hubo una fuerte separación de las asociaciones de peces de acuerdo al nivel de protección, con muchos sitios protegidos con similar composición de las agregaciones de las asociaciones que sitios no protegidos. Consideraciones finales. El estado de los peces de arrecifes coralinos y los tiburones en aguas cubanas es generalmente pobre. Sin embargo, en aquellos lugares donde las regulaciones pesqueras y ambientales se aplican adecuadamente, el estado de estas especies es mejor. Los parques nacionales Jardines de la Reina y Guanacabibes son 2 ejemplos locales de esto. Recomendamos fortalecer las alianzas entre los administradores pesqueros y las áreas protegidas marinas, los operadores turísticos, los sectores privados y públicos y otros actores para mejorar el estado de los peces de arrecifes coralinos y los tiburones a escalas de país. Muchas gracias.
Okay, so unfortunately, Fabian was not able to join us online. So at this point, I'm going to call it a wrap on the fishery session and hand over to Krista so we can start the spawning aggregation session.
Thanks, Joanna. Thanks for hanging in there, everybody. We are going to switch to reef fish spawning aggregations, which are extremely difficult to study and work with, but can be really, really rewarding when everything aligns. So our first talk is going to be Omar Rivera Garibay, and he's going to be discussing community participation in the monitoring of fishery refuges and spawning aggregations in the Mexican Caribbean.
Gracias.
Listo. Gracias por estar aquí. Buenas tardes. Me gustaría empezar contándoles que más o menos hace unos 12, 13 años, la primera vez que empecé a ir a congresos, recuerdo bien que al final de las presentaciones tenía un componente que decían que es muy importante incluir la participación comunitaria. Y si les soy muy honesto, en ese momento la verdad no entendía perfectamente como cuál era justo esa importancia. Con el tiempo ha cambiado la cosa y aprovecho para contarles que vengo de COBI, que es Comunidad y Biodiversidad, una ONG mexicana que tiene más de 25 años de experiencia trabajando en pesquería sostenible y en conservación marina desde un enfoque de participación comunitaria. No nada más yo fui entendiendo las cosas de distinta manera, sino que justamente como pueden ver, el aumento de trabajos relacionados a involucramiento de participación comunitaria en conservación marina ha aumentado. Y si queremos hablar de participación comunitaria, necesariamente entra el tema de ciencia comunitaria, de ciencia ciudadana. Y justamente, pues estamos hablando de estos procesos inclusivos y estructurados en los que cualquier persona puede generar conocimiento. Este esquema que ven aquí es de un material que generamos hace un par de años, en el que justamente damos herramientas para implementar proyectos de ciencia ciudadana. Aterrizando un poco más, me gustaría hablar hoy particularmente cómo la ciencia ciudadana se ve expresada en una herramienta de manejo como son las zonas de refugio pesquero. Así les conocemos en México. Zonas de refugio pesquero tienen otros nombres en distintas partes de Latinoamérica, ya lo hemos visto en estos días, pero a pesar de que estas herramientas tienen el objetivo de uno de los objetivos primordiales de restablecer especies de interés comercial, a lo largo de su diseño y de su implementación, la participación comunitaria es algo clave. Entonces, vamos a hablar un poco de eso en este momento. Justamente en el Caribe mexicano existen 10 polígonos vigentes de zonas de refugio activas, de zonas de refugio pesquero, y hay 8 que ya son caducados. Voy a platicarles un poco de 3 comunidades que están manejando estos refugios pesqueros, y de la primera que les quiero hablar justamente es de Punta Allen, y en este sitio hay 2 zonas de refugio pesquero. La parte de ahí arriba es San Juan y la parte de abajo es Nichejavín. En estos sitios tienen la peculiaridad de que precisamente son sitios de agregaciones reproductivas de peces. Entonces, este refugio está activo desde el 2016 y lo lleva una cooperativa pesquera justamente de Punta Allen. Nos vamos un poquito más al sur y pasamos a María Elena, que también está dentro de la reserva de la biosfera de Siancán. Y justamente este refugio pesquero, que es un complejo de 8 polígonos, es junto con otro que se decretó en Baja California Sur, de los primeros refugios pesqueros en México. Entonces tiene más de 10 años estos refugios. Y finalmente, en Banco Chinchorro, que es un poquito más al sur, está 40 Cañones, que precisamente un año después se decreta. En este momento el polígono está en renovación, pero justamente también lo llevan en este caso 3 cooperativas pesqueras las que están involucradas en su implementación. Entonces, a lo largo del tiempo lo que hemos visto es que pues, se ha generado información a través con la participación de las comunidades, de las cooperativas pesqueras, ¿no? Entonces, me gustaría platicarles de manera general qué tipo de información se ha generado a lo largo de más de 10 años de trabajo en algunos de estos sitios. Primero, hay información acerca a través del monitoreo biológico acerca de las especies objetivo de interés para las cooperativas. Entonces, hay información de más de 10 años, desde la línea base hasta ahora, en las que hemos conocido cómo es el estatus de algunas especies clave para las cooperativas pesqueras, para los pescadores. Esta foto es del grupo de monitoreo de Banco Chinchorro, precisamente, y justo, pues así como hay especies objetivo, como son la langosta, hay especies objetivo, como son peces o algunos otros invertebrados, ¿no? Entonces, hay información de hace más de 10 años, relacionado a esto. Y fuimos identificando que definitivamente es importante entender un poco variables oceanográficas. Entonces también a lo largo del tiempo hemos estado colectando información justamente sobre variables de temperatura, de salinidad, de corrientes, ¿no? Y me voy a centrar básicamente en este ejemplo, que justamente esa foto fue tomada en uno de los refugios pesqueros, es una zona de agregación en Punta Allen. Está a 31 metros este sensor y pues lo que hemos visto son definitivamente patrones que coinciden con los cambios globales, regionales de temperatura en los arrecifes. Y finalmente, respecto a la información que hemos estado generando, cogenerado, estamos hablando de que pues justamente hay información sobre monitoreo acústico en el marco de entender las agregaciones reproductivas de peces. Y mientras vemos este video, pues les platico que justamente hemos estado desarrollando monitoreo con censo visual y también monitoreo acústico pasivo para entender los patrones de las zonas de agregación en Punta Allen y en otra región en el sur del estado que se llama Xcalac. Y con esto, pues hemos tenido información que nos ha ayudado y que nos ha ayudado a entender un poco los patrones en los que estas agregaciones están sucediendo en estos refugios pesqueros. Y quiero hacer como una pequeña pausa aquí porque entendemos muy bien la importancia de la información, de los datos, pero muchas veces en estas herramientas de manejo, como son las zonas de refugio, hay información que no es tan fácil de medir o que no necesariamente es tan visibilizada. Y justamente quiero platicarles un poco de las cosas que han surgido a lo largo de estos 10 años respecto a aspectos sociales en la implementación de refugios pesqueros, ¿no? Hablamos un poco de que en el diseño de estas herramientas, en la implementación de estas herramientas, la participación es importantísima, la participación comunitaria. Y la integración del conocimiento tradicional es algo clave. Desde el diseño hasta la implementación y después el análisis de los resultados es súper crucial que tengamos el conocimiento tradicional incluido. Por otra parte, a lo largo de este tiempo, los espacios de participación igualitaria han surgido. Les quiero platicar brevemente sobre este grupo de personas que justamente es el grupo de monitoreo de Punta Allen, ahí en la Reserva de la Biosfera. Y básicamente, después de 10 años de trabajo, pasaron de tener solo 2 mujeres dentro del grupo de monitoreo a que ahora sea un grupo con mayor participación igualitaria, ¿no? Mayor participación de mujeres en la colecta de información, en todos los procesos posteriores y previos de los proyectos que están en marcha con ellos. Entonces, me detengo también aquí un poco porque justamente dentro de esta cooperativa pesquera que maneja este refugio, es el único lugar, al menos en la región, que tiene un centro de investigación comunitario liderado por ellas y ellos, ¿no? Otra de las cosas importantes justamente pues es el tema de fortalecimiento de capacidades, y de repente entendemos esto como pues capacidades técnicas, pero no solo viene de la mano con temas técnicos, ¿no? Si de repente hay interés de algunas personas, de algunas justamente personas de las cooperativas, de las comunidades en involucrarse en estos proyectos y no necesariamente quieren entrar al agua, pues hay otras alternativas, ¿no? Justamente en la que se pueden involucrar las personas a esto, ¿no? Desde temas de llenado de tanques, de pasado de información, hasta temas de difusión de los resultados que se están viendo. Y otra de las cosas súper importantes aquí es precisamente la transferencia de liderazgo y conocimiento. En esta foto están viendo a un buzo monitor de 60 años al fondo de Playera Roja, este año entrenando a un buzo monitor de 18 años en María Elena, en Quintana Roo. Entonces, estos procesos de transferir conocimiento, de transferir liderazgo, son súper claves en todo este camino. Entonces, para ir como redondeando un poco, a la hora de que hablamos de monitoreo participativo, estamos hablando que esto está impulsando ciertos procesos clave. Estamos hablando de procesos de reflexión, de aprendizaje, de concientización y de apropiación sustentable de los recursos naturales. Y aquí vuelvo a hacer otra pausa un poco, porque les comentaba que hace 12, 13 años yo tenía como esta duda de qué onda con la participación comunitaria. Y creo que definitivamente, y esta semana lo hemos estado viendo, las cosas han cambiado, han estado volteando, se empieza a voltear a ver definitivamente la importancia de incluir a las comunidades en la integración de conocimiento. Sin embargo, es buen momento, creo yo, para repensarnos un poco y para hacer como esta autocuestionarnos justamente qué nivel de participación queremos fomentar en estos trabajos, ¿no? Entonces, este trabajo viene como planteando ciertos nivel de participación y me hace mucho sentido compartirlo porque justamente existen niveles de participación en las que solamente es un actor externo impulsando la colecta de datos en las comunidades, pero hasta ahí. No se sabe más de esto, no se sabe dónde quedan los datos, no sabemos absolutamente nada desde la comunidad. Y después podemos pasar a un nivel un poco más funcional, en el que vuelve a ser un tema de que sí estamos colectando, un actor externo está impulsando la colecta de datos, pero el análisis y la interpretación de los datos está sucediendo fuera. Y después podemos pasar a un nivel más en las que ya esté habiendo un involucramiento mayor desde las comunidades. Entonces, hablamos ya de que se involucran en los procesos no solo de colecta, sino previos y posteriores. Y después, lo que deberíamos, desde mi perspectiva, aspirar es justo a un nivel más transformativo, en el que ya no haya necesidad de un actor externo impulsando estas iniciativas, estas dudas, sino que justo todo salga desde las comunidades y esto va generando normalmente líderes comunitarios, ¿no? Y este trabajo igual me hizo mucho sentido compartirlo porque hace como un análisis de publicaciones relacionadas al monitoreo participativo en Latinoamérica y tiene resultados interesantes que los centran un poco México, pero creo que nos da un poco de pistas de lo que está sucediendo en Latinoamérica, ¿no? Los trabajos de monitoreo participativo están centrados en ecosistemas terrestres. Y aquí creo que el mensaje es, tenemos mucho que aprender todavía de lo que están haciendo en comunidades con ecosistemas terrestres precisamente, ¿no? Y otra de las cosas es que pues los resultados o las publicaciones que están saliendo pues terminan normalmente en otro idioma que no necesariamente es el que es el de las comunidades, ¿no? Voy cerrando con esto porque al inicio hablaban como justo de que daba esperanza y a mí me da un montón de esperanza que de repente pues las voces de las comunidades estén viéndose en estos espacios de manera indirecta, como en este caso, o así como Gloria el lunes que nos presentó su trabajo en Yucatán. Entonces, les agradezco mucho. Cualquier cosa estamos. Gracias.
Gracias, Omar. We have time for 1 or 2 questions. Alfonso?
Sí, Omar, me gustaría saber básicamente cuál es la situación del mero de Nassau, independientemente de todo el trabajo que han hecho. ¿Cuál es el estatus de las agregaciones de desove del mero Nassau en la región donde tú estás? Y si la colecta de datos que están haciendo está sirviendo para poder saber cuál es la condición poblacional, se consume, dónde se vende. Ese tipo de información es muy importante porque a nivel regional hay un interés por esa especie.
Sí, y entiendo bien el interés, gracias. Lo que podemos saber en este momento es que vamos desde el 2018 teniendo monitoreos año con año y tú sabes bien lo complicado que de repente es bajar y observar visualmente las agregaciones, más en estos sitios, pero te puedo decir que ha habido confirmación visual desde ese año. Hemos llevado el monitoreo acústico pasivo desde ese momento y año con año tenemos información de los picos de actividad. Lo que nos falta en este momento es hacer esta match entre la actividad acústica y la confirmación visual como para tener datos más certeros de tu pregunta justamente de cómo está la población. Pero tenemos videos en este año de tanto en Xcalac como en Punta Allen de registro de más de 1.800 individuos en un buceo. Entonces están ahí las agregaciones con respecto al tema de que si se pescan o no se pescan, pues tenemos también cómo la veda está presente. Hay una veda desde febrero hasta marzo, pero si se dieron cuenta ahí en esta gráfica, los picos de actividad están presentes desde enero. Entonces hay mucho trabajo todavía por hacer y creo que aquí la voz precisamente de las cooperativas que están involucradas va a pesar mucho como en este trabajo.
Hola Omar, buenos días. Muchas gracias, te felicito. Muy innovador y muy inspirador lo que presentas. Mi pregunta es acerca de cómo identifican las comunidades, ustedes cómo identifican en las comunidades qué rol se le asigna en el monitoreo comunitario. Tú decías que había que sectorizar, por ejemplo, algunas tareas que se le asignan a las comunidades, ¿cierto? Dentro de la investigación. Todos los eslabones obviamente son importantes en un proyecto de investigación, pero la pregunta es, ¿hay algún protocolo o tú propones alguna metodología para identificar el potencial que tiene la comunidad para hacer el monitoreo? Gracias.
Gracias. Pues mira, justo en estos procesos que buscamos hacer normalmente, antes de todo hay una socialización de lo que se plantea hacer, ¿no? Y las pláticas, antes de hablar de entrar al agua, antes de hablar de asumir un rol, pues justamente nos interesa que haya como un involucramiento más profundo. Entonces, si te soy sincero, ese tipo de cosas van saliendo solas, porque definitivamente hay gente que está súper puesta para entrar al agua, ¿no? Esos son los que son más. Pero de repente notas personas que tienen interés en involucrarse, pero que no necesariamente quieren contar peces, ¿no? Entonces, creo que ese tipo de cosas se van identificando y van saliendo como sobre la marcha.
Gracias, Omar.
Vale, gracias.
Thank you. I'd like to invite Ana Giro. So going to Guatemala now to talk about the Marfish project, spawning aggregations in the Mesoamerican.
Thank you, Krista, for that introduction. I'm going to be presented, but also my colleague Antonella Rivera from the Coral Reef Alliance. So we're going to the both of us will be presenting the project. So it's a knowledge monitoring and protection of the Mesoamerican reef fish spawning aggregations. And this was centered in a project which is called the Marfish, in which we had 4 donor organizations that participated: the French Fund for the Environment, the Summit Foundation, Oceans 5, and Marisla, and 7 partner organizations across the Mesoamerican reef. So we had COBI, which we just heard, Omar. We have Tide and Sea in Belize. We had Fundaeco in Guatemala. Healthy Reefs, we are regional. The Coral Reef Alliance is regional as well, and the Mesoamerican Reef Fund, which is regional. So the project was centered in the Mesoamerican reef. We have Mexico, Belize, Guatemala, and Honduras. It's a transboundary reef, which is really important. The annual value of the reef is $4.5 million, integrated by coastline protection, tourism, and coral-associated fisheries. But one specific part of this project was also focused on the Cayman Crown Reef area, which is located between Belize and Guatemala. It's a transboundary reef there. And it's one of the healthiest reefs in the Mar, and it has rich biodiversity and a hub for reef fish reproduction as well. There. So this is the Marfish project framework, basically. So we have the objective, the main objective was also establish and monitor regional fish spawning aggregations across the Mesoamerican reef. And also the specific objectives for first was focused on across the Mesoamerican reef. And then one is in the Cayman Crown Reef itself as well. And then to promote the participatory monitoring of this network of sentinel sites. And then you can see, well, all the specific objectives and all the components. But I'm going to be talking about the component 1 and 2, and then Antonella is going to be talking about the component 3 and 4. So we'll get into that. So the component 1 was basically knowledge and protection of the Cayman Crown Reef in itself and about that specific area. So we did the diagnostics and the assessments. This was done by many of our partners too. So it was a diagnosis of the Punta Manabique wildlife refuge. This was carried out by Fundaeco. We have the ecological characterization of the coral reefs and megafauna there. That was done by us. And we also mapped and did habitat maps as well of all of this area of southern Belize, Guatemala, and also in the coastline of Honduras. So we created these habitat maps. And then for the protection, we also have the 10-year closure in Guatemala of the reef area and the expansion of the Zapodilla Keys Marine Reserve to include the Cayman Crown Reef in Belize, and then a 5-year management plan for the Zapodilla Keys as well. And there's ongoing control and surveillance. This was also supported by the project itself and also supported by the project, the Hunting Key station was furnished and equipped, and also to serve as a monitoring and patrol station of the reef, because it's the closest by of this, the Cayman Crown. And then the financial mechanisms, we have the financial strategy and action plans for the Cayman Crown management in Guatemala and Belize that was established and was set, and the co-financing for Cayman Crown protection as well. And the increased endowment funds from the initial, well, we had this initial 507,011 in 2019 to 1,125,100 in July. And this is, the endowment fund is through the Mesoamerican Reef Fund. And then, well, the component two was the Sentinel Site Observation Network. So this was in the whole entire Mesoamerican Reef. And we did the FSA monitoring. This was carried out also by the partner organizations that I mentioned. We have the FSA regional monitoring protocol established. It was standardized, all the protocol between the four Mesoamerican Reef countries, and also the monitoring network of 10 multi-species FSA sites and Sentinel sites that were selected. I'm going to talk a little bit more about the selection process and what changed, but we started with those. We trained 19 participants on standardized techniques for FSA monitoring, provided 47 equipment units for FSA monitoring. We tried to establish a network of acoustic receivers, the hydrophones, basically, in many of the countries, supported the training of 152 participants in coral reef monitoring. And this was done by us. We trained the participants, but also did the monitoring near also the FSA sites. So what's going on in the reef sites where we have, for example, the groupers and snappers, and we did that part and then complemented it with the monitoring in the FSAs per se. So we had the data and knowledge sharing. We had a report. We developed the regional FSA data platform, which is hosting 569 regional FSA data surveys. So everyone can upload their data, they can share it, they can keep it to themselves, but we keep that as anyone that uses the platform can actually decide, because we know that FSA sites are, well, kind of vulnerable, so we sometimes don't want to share the data, but we share it between the region. And then generated 8 documents of FSA analyzing all the different FSA sites and what's going on, the status, and the reports and policy. We did 2 policy briefs as well and 3 scientific publications. So this is how the Sentinel sites changed over the period of the project. We have the COVID, the pandemic in between the projects, so that, well, also took longer to finalize the project. But basically what I wanted to show here is also what Omar was saying. The FSA site sometimes is very difficult to monitor. And so we had to change some of the sites that either because they were, so difficult to get to, or the conditions there were always really bad, and we couldn't collect the data. So as it started, we had all these different sites in Belize and in Mexico and Honduras, and then how they all changed. And then we had the Cayman Crown Reef area where we didn't know where the FSA sites were, so we had to do all that previous monitoring for that part as well. And Antonella, I'll leave you.
Thank you, Ana. So I will be talking about the final 2 components. And in this one, in promoting the social acceptability of protecting spawning grounds, what we're looking for is really to raise awareness on why FSAs are important and their benefit, to understand and mitigate the impacts to livelihoods that they can have, and to gain support and empowerment of communities so they're part of the process. And we did this in 3 different ways. One is communication and outreach. The other one is livelihood diversification, and the third one is capacity building. So when it comes to communication and outreach, a long video was developed to really detail what the Marfish project is, but then 6 small tidbits were created. So those can be shared on social media and everybody can see what's happening and what are more suited to their needs. And we also supported the development of a documentary called Treasure of the Caribbean, developed by Ana Salcedo, and that was shared with national decision makers. So we worked with the government, but also in all the different fishers communities. So they could also be part of the process and understand what happened. As Anna mentioned, we did policy briefs in 2020 when we started, but also when we finished the project, so they knew what the advances were. And finally, we also support it with the report card, which you heard about earlier this morning. When it comes to livelihood diversification strategies, we wanted to be very holistic. So the first thing we did was a review of 17 communities in the Gulf of Honduras to understand what their status was and how the closures could impact them. And then we did follow-up workshops with the fishers themselves to understand what these challenges were and to co-design solutions. And both of these are published in scientific journals. And with this information, we developed pilot projects, 3 in Honduras, 1 in Guatemala, and a whole feasibility study for 3 communities in Belize. And finally, when it comes to capacity building, the first thing we did is once again start with awareness. So we had workshops for 48 fishers all across the Gulf of Honduras. And then we started working with capacity building. So more than 70 fishers in Honduras were trained, as well as over 450 fishers in Guatemala. And with this information, then we did a whole regional exchange. So whatever we learned, let's share it with the rest of the communities. The final component is pretty much what you would expect. It's project management. It was a little bit harder when we're considering so many partners in so many countries, but we held continuous strategic planning meetings, obviously financial reports. And in the last year, we had a visit to ensure evaluate how the process went and what the successes were. And it was so successful that we've been invited to develop a note of opportunity to build on the results of this project. And because we're close on time, this is basically the project implementation timeline. As you can see, it went on for 6 years, so it was a bit of a longer process. COVID came in. We also had a lot of weather constraints. As you know, sampling FSAs isn't easy, and we do depend a lot on how the weather is doing. But despite all this, we were managed to meet all of our objectives. Fisher's Cayman Crown was protected very early on in 2020, and it continued to be monitored. Fishers were also included in this process, and a Sentinel site network was established, and many of them are still being monitored today, even after the project has developed. So we built that capacity locally in each country. And finally, the community has been aware of these new areas, and they are very adamant of supporting these initiatives. We had a little video here, but I'm going to skip it because we're close on time. So you can see this video if you scan the QR link for the Marfish webpage, and you can see all of this outreach material I was just telling you about. Thank you.
Thank you, Anna and Antonella. Does anyone have any questions? Alfonso.
Thank you. Thank you. Which are the top 5 species that aggregate to spawn, and what's the situation of the aggregation of those top 5 species in the region?
I could start with Honduras if you want. So we really only saw groupers aggregating in Honduras. And it was interesting because we have a few sites which are exclusively tiger grouper. And then we have our Keystone site, which is a multi-species site, which has tiger grouper, black grouper, yellowfin grouper, and Nassau grouper together. And the status is they are very much overexploited from what we know and what we're seeing. We're seeing abundance was going up, but fish biomass is going down. So we're getting more, but they're getting smaller. And that's why we've worked with the government to develop new policies, especially in Honduras, where we have a new closed season for all these species I mentioned from November to April, both inclusive. And Nassau grouper has been banned in the country since 2017. So fully banned all year long for Nassau, and then a temporal ban for the rest of the species.
Yeah, and I think the patterns are pretty much the same also in the parts of of the Cayman Crown area and in southern Belize where we had like part of the Marfish project as well. But yeah, I mean, we have seen the amount of fish go down, and yeah, basically they are being, I mean, we've been implementing regulations, but I think, yeah, we need to implement more enforcement in these specific areas.
And just keep getting the communities involved as well, because they know of it, but they're still trying to feel where they can become part of the process as well. So we're still trying to do that too.
We have time for one more quick question if there are any.
I had
Oh, so sorry. Didn't see you, Alejandro.
Great presentation. What kind of enforcement are you trying to implement or try to work with the authorities to oversee in those protected areas or spawning aggregations?
I can start really quickly just with Honduras. So we're working with the local NGOs. We always work very closely with co-managers, and they have patrol boats going out everywhere to enforce these regulations. But it's not everywhere. So we're protecting the FSA itself, but not when the fish migrate to come to the FSA. And that is why we're working with the national government and the fisheries department so they can start enforcing that as well. And then for Cayman Crown.
Yeah, for Cayman Crown, since it's a transboundary reef, it has been a bit difficult, that part. But yeah, we have the authorities also in Guatemala and in Belize protecting, you know, that area. What we established in Guatemala is a no fishing ban for 10 years, but we're, you know, that ban, you know, is going to end pretty soon. So yeah, we want to keep on implementing those regulations. And for the Zapatela Keys Marine Reserve, yeah, they have their station in Hunting Key, and they have had the support with this project, new equipment and like implementing the surveillance, even like with high technology in the key. They have, you know, some really high tech where you can actually see if the boats go by or are going to Cayman Crown. So yeah, trying to work with them too.
Thank you, Anna and Antonella. We don't have any time for questions, so you can follow up with them after. I'd like to invite Janelle Layton. She's going to be talking about energy output as spawning aggregations for tiger grouper in the Cayman Islands.
Hello, good morning, or afternoon. It's almost 1 o'clock. I am Janelle Layton. I'm one of the students on the Grouper Moon project, based in Little Cayman Islands. And this is one of the most successful fish conservation stories in the Caribbean. And it's primarily focused on Nassau grouper, but we also study tiger, yellowfin grouper. And so my presentation is really focusing on energy output during spawning aggregation events of tiger grouper on Little Cayman. Some background on tiger grouper: they are a large reef predator found in the Western Atlantic Ocean, specifically the Caribbean and the Gulf as well. And they are ecologically important as large reef predators because they can help structure coral reef food webs. Outside of that, they form fish spawning aggregations, which is not only a really cool life replenishing event, but it's a form of reproduction that releases massive numbers of gametes into the water column, forming the base of a marine food web. And these nutrients can support other organisms like plankton, invertebrates, other fish species, fueling energy flow and transfer through an ecosystem. These spawning events can even attract large predators. I would like to note this is a Nassau grouper spawning aggregation, not tiger grouper, but the video is cool because you actually see a large predator come in, a successful shark attack, and having these aggregations really supports concentrated energy transfer and broader diversity and trophic interactions. And I keep bringing up energy because it is extremely important. It's the currency of the ocean. Since the 40s, it's been cited that everything in community dynamics has been driven by energy. We see this from abiotic to biotic factors, vice versa, and then through the food web as well. And energy in general will just play a crucial role in maintaining an ecosystem's balance and productivity. So knowing the importance of tiger grouper and energy, unfortunately, the species is data deficient. We just don't know enough about them to classify them on the IUCN Red List. But we do know closely related species like Nassau grouper are on the spectrum of critically endangered. As I said, it's a spawning species on Little Cayman. They spawn on the west end of Little Cayman. And this happens during winter months around the full moon. Tiger grouper will generally spawn a few days after Nassau grouper begin. This is a shared aggregation site, and we also see yellowfin, black grouper, tiger, Nassau, et cetera, at this site. And this all ties into my research objective, understanding energy output during these aggregation events of tiger grouper. And to do that, we collect samples when the grouper are spawning. In a second, you'll see a spawning burst, and a diver will immediately go after the spawning burst with a butterfly net to collect any embryos that were released into that water column. This is Dr. Ali Kandomo, one of the senior scientists on the Grouper Moon project. And then that butterfly net will go into a gallon-sized Ziploc bag on the dive. This is Dr. Scott Happel, my current PI, and that Ziploc bag is pre-labeled, so we can actually go back and identify which specific spawning event those embryos came from. Then all those embryos come back to a lab. On the right, you'll see a picture of what these eggs look like or embryos look like zoomed in with just a phone camera. On the left, you'll see a picture of what it looks like under a microscope. And I'll weigh a set count of embryos in the lab. So I'll count like 60 embryos and get that weight to get a wet weight. And then overnight, I'll dry them and then reweigh them to get a dry weight so we can get a wet weight to dry weight ratio per individual embryo. From there, it goes through this process called bomb calorimetry. If you're not familiar with that is, it's essentially a way that you can measure the amount of energy, specifically calories, it takes to blow up an item. And so this item happens to be a bunch of dried up eggs turned into these pellets. And we do that to get the caloric content of a single pellet. We did this for 1 night of spawning in January 2023 and 4 nights of spawning in February of 2025. I would like to note the Grouper Moon project primarily focuses on Nassau grouper. And so in January 2023, tiger grouper was very much an afterthought, and that's why there's only 1 night of spawning. And then after what we found out in 2023, it was really interesting to expand, and we did look at more samples in 2025. So one of my first research questions was pretty basic: how much energy is in a single tiger grouper embryo? And the answer was 0.011 kilojoules, which may not mean much to most people. So to make it have more relevance, the average human consumes about 8,300 kilojoules a day. And if you're like me trying to do a bunch of math, that is over 760,000 embryos to feed a single human in a day. But how many embryos, how many eggs does a single female release during a spawning event? And so that leads to my next question of how much energy is produced in a spawning event. To get that, we needed batch fecundity estimates of tiger grouper, which don't exist, but using a paper from Collins et al. In 2002, a closely related species, red grouper, was studied to get batch fecundity estimates. And then we were able to use length data from the Grouper Moon project to get those estimates. And a little over 137,000 eggs are produced from a single female in a single night on average. And so that equates to about 1,500 kilojoules in a single spawning event, which could feed a human for about 4.3 hours. But knowing that, how many females are actually in the water and then how much energy is going out in an entire spawning night. And that is still a mystery. As I said before, tiger grouper are data deficient. We just don't have the population estimates over time for tiger grouper when they are spawning. But that is a future question of interest and what I would love to get those population estimates to figure that out. My next question was looking into, does energy output vary between closely related species? And I started to answer this in 2023. On the y-axis, we're looking at energy content per embryo, specifically in kilojoules at wet weight. And on the x-axis, we're looking at different spawning bursts. On the red, we'll see Nassau grouper spawning on night 1. Green is Nassau grouper spawning on night 2. And blue is tiger grouper spawning on night 1. And we did see a significant difference between night 1 of tiger grouper and night 1 of Nassau grouper, emphasizing that there is energy or differences in energy allocation per species. And then we wanted to look into energy output between different spawning nights and different spawning years. And in 2025, when we had more nights of spawning for tiger grouper, we were able to compare energy content per embryo over 4 different nights, and then also energy content per embryo by the year 2023 and 2025. In both situations, we did not see any significant differences over time. I would like to note, we do have inconsistent sample sizes. 2023 to 2025, like I said before, it wasn't a priority in 2023, so we didn't have as many collections. And then across spawning nights, night 2 specifically, we just didn't see as many tiger grouper in the water and didn't get as many eggs as we would like. And then lastly, I was looking at energy output varying between different spawning events. And in this figure, we're looking at energy content per embryo versus spawning night 3 and spawning night 4. And then each different shade of blue on spawning night 3 represents a specific spawning event in that single night. And then each shade of red represents a different single spawning event on that night. And we did notice that there was variability. Significantly, we did see a significant difference in night 3 and another one in night 4. And so although we may not see significant changes over different days combined or on average, we do see individual variability within a single night. In summary, energy is important for various reasons I mentioned before, and closely related species can vary in energy allocation, which really emphasizes the need for species-specific management when trying to protect these spawning aggregations. Also, there are little differences in across spawning nights or between years for tiger grouper, and so sustained conservation efforts throughout the entire spawning period is also critically important. And then energy dynamics can change with fish size or fish length. It'll impact different fish sizes will have different fecundity estimates. And so that is important to consider when you are studying these aggregations in the future. So not only thinking about the size of the aggregation, but the health and size of the fish in the future as well. And in the future, I'd like to get tiger grouper population estimates to further expand this study. I would also like to better analyze differences in variability and not just significant differences in means or medians. And this is a shared aggregation site, so my next target species is yellowfin grouper, if I can do that next or in a couple of years, and apply a lot of this information into community and ecological context. Do we see bottom-up control of large reef predators having a significant impact? Do changes in energy output affect predator-prey dynamics during these aggregations? And do changes in energy output impact nutrient availability in the water column as well? I'd like to thank this whole list, specifically GCFI, for the Outstanding Student Achievement Award last year, which allowed me to come back and present this work this year, and the Grouper Moon Project and all of the organizations tied in with Grouper Moon. Thank you.
Thank you, Janelle. We have time for questions. Will?
Thanks so much. That was a great talk. And I want to, again, congratulate the work that's been done in the Caymans in terms of the protection that's led to kind of exponential increases in Nassau, where everybody else, is declining. So kudos to you guys. It's not just science. We got to have boats on the water, making sure that fish stay in there. So just a side comment there. But the question has to do with the energy output regarding how comfortable are you with the extrapolation from red grouper fecundity to tiger grouper fecundity and output. And the corollary, of course, is, do you have any plans to measure that directly in tiger?
Yeah, I am fairly comfortable with it right now in the sense that it's one of the only options I have to get fecundity estimates. I would like to, actually the goal is to do it for Nassau grouper first before I do it for tiger grouper. It's not, I mean, that is the plan in future work. However, I just can't do it right now. I don't have the capacity. Yeah.
Thank you. Henry?
Hi, Janet. Nice presentation.
Thank you.
Have you or anybody looked at variability in egg quality and how that may link to, energy content of the eggs? Can that be used as a proxy for the condition of the females? So that's my first question. And second of, have you guys looked at how much energy that represents for the female in terms of her basic metabolic needs? And does that change with female size?
Sorry, the second question you're asking how
So the
much?
first question is about egg quality variation.
Yeah, egg quality, yes.
Has anybody looked at that?
So I'll go in the first one. Yeah, there are different studies looking into like lipid content of eggs versus like fatty acids, et cetera, that are trying to quantify the quality of eggs of different species, not specifically for tiger grouper, but primarily Nassau grouper and yellowfin as of right now. And then your second part of the question.
So I guess the first part of the first question was, is that, energy output linked in any way to egg quality?
Oh, the energy output? No, not yet. We haven't gotten that far.
And then in terms of basal metabolic needs of the female, how much energy does it require for it to produce, what percentage of, how does that affect the female in terms of condition to produce that energy output?
Yeah, that's something we haven't looked into yet. And so the part of my future work, I'm trying to get those like broader ecological and community impacts, that's part of like the future questions I would like to answer.
Okay, thank you. Unfortunately, we don't have time for any more questions. I have a question for you, so I'll find you later.
Okay.
But we're going to switch to the last presentation so everyone can go get lunch. So we have Joanna Pitt. She's going to be talking about management measures and how that's helped with spawning aggregations, I'm assuming in Bermuda.
Good afternoon, everyone. Thanks for hanging out for one more presentation before lunch. So this presentation was kind of a little bit more aimed at the previous session, but numbers meant that, and the fact that it includes spawning aggregations means that I'm talking in this session. So this work it's pulling on a lot of long-term data that we have in Bermuda, but we could not have done it without our partners at MLab and with the support of the Waite Institute, who are our major partners on a big project we've got going on at the moment, which is the Bermuda Ocean Prosperity Program that includes a marine spatial plan and blue economy stuff, but also this key fisheries thread that I'm going to be talking about today. So I'm going to be talking about a range of management strategies and then what we've done to kind of, see if they're actually working. So most of us in this room at this point know that grouper species and a lot of other marine species are vulnerable to overfishing because of certain life history traits that they may have. And basically, a lot of the groupers tick all three of these boxes that, they're long-lived and they mature late. Very often they're protogynous hermaphrodites, and a lot of them aggregate to spawn. And so they need careful management. But a lot of the management approaches that we're encouraged to take need a lot of data and a lot of resources to enforce. And so our question is, can simple management strategies like the ones that we've been doing all this time, can they yield similar benefits? And the specific measures that Bermuda's been able to put in place, we now have a minimum size for retention. We prohibit fishing when the species are most vulnerable, i.e. when they're aggregating. And we have been among a few places in the region that have restricted what I'm going to euphemistically refer to as non-selective and destructive fishing gears, i.e. fish traps. So we've done all this, and we've had different extents of buy-in from different parts of the community. Is it worth it? Are these things working? And so I'm going to be working through this case study of the red hind, for which we have over 50 years of management history. And all of this has been working with what we knew at the time, because 50 years ago, we didn't know that much. And it has been in partnership with fishers, because you get nowhere if you don't start there. So the main goals have been to protect reproduction and to also kind of make sure the catch is not too much, for want of a better way to putting it. And we've been focusing on measures that were, at least for us, easy to implement and enforce, i.e. we had some data on which to base them, and it wasn't going to need a massive team for enforcement. And the bonus is that actually these measures are, for the most part, at least easy for people to understand and relate to, and that in theory sort of promotes better compliance. And so it was actually the commercial fishers in Bermuda that were the first to really recognize how vulnerable the spawning aggregations were, and they suggested that they should be closed to some or all fishing. And this goes back to the early 1970s. And so with the input of the fishers, the spawning aggregations were closed during the main spawning season with a buffer period. And in the early to mid 1970s, this was a pretty novel thing. So even though it's now considered best practice, this was the first example of its kind. A lot of things have changed during this time, however, and management has had to adapt. So what you're seeing here are the first two known aggregations, and they were protected with this triangular line of sight protected areas from the 1970s. In 1989, a third area in the Northeast was also protected. Coming online in 2005, we discovered a black grouper spawning aggregation where that little asterisk is. And so the two northeastern areas were combined into a single area delimited by GPS coordinates. And then the southwestern area was refined similarly in 2012. And so this little graphic shows you our landings history over time. And so what we can see, starting in when we first started collecting fisheries landing data in 1975, things are not looking good. Fish pots are everywhere. And you can see we are on a downwards trend. There's a little dip and then a recovery in the early 80s in landings, recovery in landings. That was the first attempt to regulate fish pots. And so the fishers were allowed fewer of them, so they made them bigger, and the catches went back up again. Moving on to 1990, all the reductions are not working, and so fish pots are banned. We also implemented at that time commercial and recreational bag limits to support the trap ban. And so during the spawning season, there was a bag limit of 10 fish per boat per day. In 1996, we implemented a minimum size limit of 35 centimeters. And that was based on us being able to do size at age based on otoliths. And you can see the changes to the spawning aggregations that I talked about previously, adding, changing the closure time, closure areas. Then in 2014 and 2016, we saw the April catches of red hind go through the roof. And it's like, what is going on? And basically the fishes are now aggregating earlier because of climate change. And anybody who was in Merida in 2017, you hopefully saw my presentation on that. So from 2017 onwards, we shifted the closure period forward by 2 weeks. So the aggregations are now closed from mid-April instead of from the 1st of May. And we made, at that time, the 10 fish per boat per day bag limit for recreational fishers was made year-round. Commercial fishers then got a bag limit of 50 fish per boat per day during the month of April, and then remained at 10 fish per boat per day for the rest of the summer, but no limits the rest of the year. So this is now what we have in terms of current management measures. I'm not going to repeat myself. Hopefully everybody remembers what I've just been talking about. And so we've got positive signs. There's if you looked at that graph, there was an increase in landings from 2012 without a lot of added effort. Juvenile red hind are showing up in surveys, and there's been an increase in average fish size, both at the spawning aggregations, and you can see how between the 70s and 2003, we've got an increase in the average size of the aggregations from about 35 centimeters to almost 48 centimeters. And we've got an increase in the mean size landed by the commercial fishery from 1997 onwards, when we put the minimum size restriction in place. So we're going up by almost a pound in terms of the average fish landed. So how do we actually evaluate the stock status? It's like, we have these nice signs. It would be nice if we could say, yeah, so we actually have a number that we can put on this. So we look at what data we have. We do have the life history information. We have good information locally, but there are also data from other nearby locations, nearby being a relative term for Bermuda, obviously. We have our long-term commercial landings data. We have historic and newly collected length frequency data, both fishery independent from targeted catch and release at the spawning aggregations, but also collected from the commercial fishery. And these types of data are useful in modern population modeling methods, catch depletion approaches, but also ways of evaluating spawning potential as a proxy for population status. So we did use the fisheries landings data for a depletion-based stock reduction analysis. And this is kind of, it throws you out a theoretical maximum sustainable yield. And, it gave us a number. And it also estimates the current biomass compared to the theoretical unfished biomass. And it gave us an estimated exploitation rate of about 5%. And, MSY is, ideally happening at about 14%. But if this is an overestimate, which part of me kind of feels like it might be, it does risk promoting overfishing. And not everybody has this kind of data. So using the length frequency data through time and having a look at the spawning potential ratio seemed like a great option. So we have all these historical data. This is using the ones that I showed you previously from the spawning aggregations. And then we have another lot of data from the commercial fishery. And you can see that it's got a slightly different profile compared to the others. And what we can see here, we can see the minimum size. This is, so the minimum size is the vertical red bar, and you can see when it was implemented, and you can see a change in the mean size in all of these little population distributions and how that changes once the minimum size limit is implemented. And so we did a length-based spawning potential ratio that included lengths from approximately 200 fish per year with life history parameters. And the important thing to say is that the same fishing method should be used for all measured fish. The model simulates the length frequency distribution of an unfished population and compares your observed length frequency to the model, and it gives you basically an idea of the current spawning stock as a proportion of the unfished stock and reflects the status of the population. It also calculates the selectivity of fishing gear on different size classes. So this is busy, but we're getting to the exciting stuff now. So again, if you recall all the demographic profiles, most of these little dots were getting darker through time. And then the black one on the end is the one from the commercial fishery. And so we can see that we've got spawning potential ratios at equivalent to about 40% and 50% relative to an unfished stock from the spawning aggregations. But it's also nice to see that we're getting that from the commercial fishery again as well. So basically what that translates to in management terms is the population is not overfished. We also see increased selectivity for bigger fish over time. As the lines get darker, that's progressing through time. And we can also see that sampling is more selective than the commercial fishery, which is important for the next part. Because the apical F over M, which is basically how does fishing mortality compare to natural mortality? It looks really high at the spawning aggregations. But again, if recalling what I just said, the fishery is actually less selective than the spawning aggregation. So when we look at the data from the commercial fishery, we see that actual fishing mortality is 0.7, i.e. less than 1. So that tells us that overfishing is likely not occurring. So there are lots of caveats on using these methods for protogynous and aggregating species, but we can sort of say, based on what we have, that these management measures, despite being kind of like your basic stuff, have reduced catch rates in general, and especially at the aggregations. They've increased the mean size and weight of landed fish and spawning fish. They've improved the spawning potential and population status of the population, with the SPR indicating that it is not overfished. And we can also say with reasonable confidence that current commercial fishing practices are likely sustainable. The harvest rates are low relative to the theoretical maximum sustainable yield, and that comes from the depletion-based stock reduction analysis. And fishing does not overly target a particular size class, and that comes from the apical F over M. The data do further highlight the risk of fishing spawning aggregations because how they can overly target particular size classes. But I feel confident standing before you and saying that simple management measures can be effective. And all of these measures have likely contributed to the apparent success. But regular reevaluation and adjustment is important. The other thing for other people, you don't need lots of data to take useful action. Use the information that fishers are prepared to share and the data that you have and work together. Get more data if you can. The length-based spawning potential ratio method doesn't actually require a lot of data. You can get it from the commercial fishery, and even 1 year of data can give you something useful. And every year extra is just adding to what you know. And it can also be used to compare the impact of fishing gears using that apical F over M thing. It's also good to find partners that can do the math. I need to shout out to Michelle Burns, who did a lot of the calculation and modeling for this. But there are also online resources available, the fisheriestoolkit.org that Bill Harford mentioned the other day. This was actually in my presentation before he talked about it. I'll just say that. But again, regular evaluation and adjustment is important. Thank you. Wow.
Thank you, Joanna. Unfortunately, we do not have time for questions, so you'll have to find Joanna during lunch or one of the coffee breaks. I'd like to invite the presenters that are still in the room from the previous 2 sessions to take a group photo, and everyone else enjoy your lunch. And Fadila has an announcement.
If anyone has lost a jacket, this was left behind at Cinefish last night, so we'll keep it in the. It's yours, Mike? Okay, I think he just has his hand up. Also, we're going to start promptly at 2 for this afternoon's activity, this afternoon session. And tonight's social is in Teatro. I don't think anyone's listening.
And I'm going to grab the mic for one more time and give a final plug for the silent auction. You have your lunch break and this afternoon's coffee break to check your bids, increase your bids. Make sure that you have a silent auction item to take home with you, because we don't want to take them back home with us.